Okay, hearing none, I will let Hudson take over and give you the pearls of wisdom around environment of care. Thank you, Hudson. I'll
Yep.
see you
All right.
guys soon.
Thank you,
Bye.
Tanu. Good evening, everyone.
Awesome. So this week is one of my favorite topics, which is environment of care. So this is really around everything that is not going to directly touch the patient, but in so many instances has a lot of impact to the patient, whether it's the building, the environmental surfaces we touch, the laundry. So it's a lot of areas that, frankly, I think infection prevention and control touches routinely, but doesn't necessarily get credit for. So a lot of people, when they hear the term environment of care, they automatically think, oh, it's cleaning and disinfection. I think by the time we get done with this hour, you'll say, well, this is a lot more than that, and it gives us a little bit of a broader scope. So we'll talk a little bit about sort of what safe environment, realizing that we can't make everything risk-free, right? Everything in healthcare has some type of intrinsic risk associated with it. So we'll talk a little bit about water, laundry, waste management, cleaning, disinfection, what that cleaning disinfection looks like in terms of EPA regulations. We'll talk a little bit also about how to evaluate cleaning and disinfection products and how we should do that from the IP lens. The more that you can get involved in that process up front, the better, right? Because if you're involved in the selection of the products, guess what? You make sure that it's going to be the right product for the right role with the right individual, and also the right training is taking place as well. So it just makes our life a lot easier. Then we'll move into a little bit about sort of that recall management perspective, which is pretty big right now. I think last year, I don't quote me on the number, but there were about 7,000 recalls that came out through the FDA that impacted something related to an adverse event related to a medical device. Part of those could be related to infection prevention. And so, whether it's contamination like sterilization failures or food failures or some type of medication failure, or supplies that did not function as they should, those can all have direct impacts to our world. And then we'll look a little bit at environmental pathogens like Legionella or Aspergillus and finish up with a little bit of everybody's favorite topic, which is construction, renovation, and maintenance. So here's sort of how I break that down as we go through. So we're going to start with HVAC. And what I will say here is number one is you need a well-functioning HVAC system. That is a big ask in healthcare, especially here in Georgia, because what this morning, it was what 40 degrees or something like that, and it's going to be almost 80 degrees. And so we have this like ebb and flow of the temperatures, the humidity, the pollen count. And so not only are we relying upon a well-functioning HVAC system, but we also want fresh air exchanges. So, generally speaking, for our airborne isolation rooms, we want greater than 12 air exchanges per hour. And we also want two fresh air exchanges coming in from the outside. So, again, if we're in California, right, this may be a different conversation because fresh air exchanges, when they have wildfires, can be a whole different arena. And so remember on the exam, they're going to only ask you general questions. They're not going to ask you anything that's specific to Georgia or California. It's going to only be what the standards say. So if we're talking about general air exchanges for airborne isolation, it's going to be 12 or more. Fresh air exchanges, it's going to be greater than two. One of the other things that we're looking for too is how well do we maintain it? Now, the hospital typically does not maintain its own HVAC system. That's normally done by an HVAC contractor. So, as an IP, right, we want to know who the contractor is, what their level of training is, and then also how often do they come out and do the service and the maintenance, and what the records are associated with that, particularly for something like Legionella, right? If we find out, well, wait a minute, they haven't serviced the cooling towers in the last 2 years. Well, that's helpful for us to know that, especially if we have a potential Legionella outbreak. And another thing that we're concerned with HVAC systems is not just mold growth, which is pretty common actually, but more biofilm. So anytime I've got a area that is not exposed to light, it's confined space, and I have potential for water or moisture, I have biofilm and mold that could grow in there. And so it's really important for us to regulate and make sure that those ducts are also cleaned as well. So the ASHRAE 170 standard is definitely something that you want to know because that is what guides us for airborne sort of isolation as well as what our minimum air exchanges are in different types of environments. The one that typically does come up on the CIC exam is the operating room, because obviously that's a high risk environment. The air exchanges at a minimum per hour for that are going to be 20, and outdoor air exchanges are a minimum of 4. And that's going to be a positive pressure environment. So, because of the risk associated with that, we want to make sure that's also HEPA filtered there. Now, let's take a different example, like a general patient care room, like a med surg room. Much lower, right? We could look at a minimum air exchanges of 6 and an outdoor air exchanges, like we said earlier on the previous slide, of 2. I want to go to another example, though, that's particularly relevant, which is a bronchoscopy suite, right? Because if you're doing sputum collection, you can have aerosolization. So that's why you see that as a much higher, just like an isolation room or protective environment. So you're going to be looking at a minimum air exchange of 20, or I'm sorry, 12, and then a minimum outdoor air exchange of 2, and that's going to be negative pressure. Keep in mind that sometimes bronchoscopy may be done in regular traditional operating rooms, and so you may actually have more air exchanges associated with that. The pharmacy, though, can be separated into the sterile and the non-sterile area, and this is another potential test question that comes up. As a matter of fact, it was on my exam years ago. It asked me about, in the sterile compounding area. So that's going to be 30 plus air exchanges again because we're mixing product in a sterile hood, and that's going to again be positive pressure. So let's transition and look at water pathogen management. So CMS, which is Medicare and Medicaid services, do require us to have a water management program. Now, that does not mean that the IP has to do it. It means the healthcare facility has to do that. So most often, this is going to be led by facility services, engineering, whatever department you call it, but it's going to be under the ASHRAE 188 standard. And the great news is CDC actually has a very robust, comprehensive, and fully customizable water management toolkit on their website that you can use. And so the sort of 5 steps that we're going to follow, the very first thing that we need to do is figure out what our water system is, right? So every healthcare system is different. Every sort of building is different, the way it's designed. Some are newer, some are older. We may have a combination of all of the above. The second thing we're going to do as the IP is figure out what our hazards are within that existing water system. Again, that should be done by campus. So if we've got 6, hospitals, every one should have its own water management program. Now, we could apply general principles across an entire health system, but every campus is going to have its own specific needs. The third thing that we're going to look at is control limits, right? What are we accepting? What are we not accepting? And then how are we going to manage those intricacies? Especially if we're doing construction, renovation, moving things around. We've got some specific issues that we've identified as an example. And that leads us to more of the continuous quality improvement parts of the process, which is 4 and 5. And so that's going to be your monitoring and responding, and then specifically looking at validation and verification. If I don't know what I'm looking for, I can't inspect it. And so you inspect what you expect. So that's where the monitoring and verification comes in. So I may have to send water quality samples off, for example. I may have to get laboratory testing to ensure that, yes, it's actually working. Now, a sort of bad example that's happened to me several times is I've gone in a hotel and I'll go to the down the hallway on the floor and I'll look at the ice dispenser and I'll come back very disappointed because I'll open the ice machine and you'll think, dear God, when was the last time this thing was cleaned? Because when you open it, you see the visible mold. Now, luckily, that very, very rarely, if ever, happens to me at major hotel brands like Hilton or Marriott. And the reason is that they have a water management program in place for all of their properties, including filtered water that comes into the device. They're supposed to clean them out every single week. So sometimes you'll see the ice machine is out of order, and that's for that reason. But in the very back, you'll actually, if you look in the back of the machine, you'll actually see like a box and you'll see a filter that comes down with the water, and that's for that reason. So the areas that we're most concerned with are going to be just that, the ice machines, your water dispensers. Unfortunately, anytime we have some type of decorative fountain or any type of water feature, those are always problematic for us for many reasons. Our cooling towers are certainly huge. And then if you have some type of physical therapy or occupational therapy, if we're doing like hydrotherapy or hot tubs, and then another area that may be both inpatient and outpatient is going to be our hemodialysis water systems. So, especially in the outpatient environment, that is primarily managed by DaVita and Fresenius and DCI, but we all have inpatient dialysis too. So, are we properly managing the water systems associated with that? So the program is going to call for us to have a written program, right? We have to have a plan in place that is, again, customizable for the facility. We need to have a trained team, which consists of multiple experts, infection prevention, building services, microbiology, infectious disease, right? We may have an external expert that helps us with that. We then have to look at what is our system and our infrastructure to say what's going to be the potential challenge associated with that. And then the last thing that we have to do is identify what our results are and then decide what our corrective action will be. So if I come back with specific pathogens, how am I going to take care of getting those out of our system as an example? So Legionella is definitely one of those areas that we're certainly going to be concerned with, right? And obviously we want to ensure that we are managing this proactively. We all probably recall when the downtown, I think it was the Hilton, had Legionella. As a matter of fact, I stayed in a hotel when I got off the plane in Newark, New Jersey. This was about 2 years ago. There's a big sign that said this hotel is closed due to Legionella. And I thought, Lord, is this my hotel? Thank God it wasn't. But then when I checked into the hotel next to it, it was managed by the same company, right? And so you think through your head, well, wait a minute, how are these hotels managed? How are these properties managed? What about, the hospital? How are we doing that? So if we think about the pathogen itself, it's a gram negative, right? It is very, very hardy. And so it can survive in a wide variety of temperatures, particularly in biofilm. And so when we have residual left over in these conduits, that's when we unfortunately get that biofilm formation, especially with aerosols, which is where we get the Legionnaire's disease. The good news is it's not person to person, so it's not communicable from that perspective. But the challenge for us, especially with our immunocompromised patients, is that's where they're going to get that Legionnaire's disease, aka the pneumonia. So, how do we actually manage it? Well, certainly get hot water or certainly maintain your cold water. There are different technologies available, like UV disinfection is particularly common and very effective. Chlorination is certainly out there and has been out there for years. And then copper or silver ionization is another one. One of the best things we have to do is just flush. And so this is something that we want to do routinely to make sure we don't have any sort of stagnant water in the system, particularly for maybe a unit that's not been used, or if we have areas that might be shut down, or maybe an area that doesn't have a lot of throughput that's not flipped over, right? And so we're going to test those water supply areas at a minimum weekly in those high risk areas, particularly like bone marrow transplant, oncology, burn units, and things of that nature. What if we have an outbreak, though? Very first thing we're going to do is confirm that it's real, right? We want to make sure that that diagnosis is actually real. And there's different ways from a laboratory perspective for us to do that. If it is real, then of course we want to get our team together, right? So infectious disease, patient safety, risk management. You want to get certainly infection prevention, your nursing team, your providers, pharmacy, and you're going to have to get your legal and public relations team together because at this point, this is going to go public. And then, of course, we're going to do our standard epidemiologic investigation and identify our cases, right? We're going to do that through our line listing, collect our water samples, look at what remediation needs to be performed, go ahead and institute that, right? And that may include moving patients and things like that. We're going to work with public health, right? Because public health is going to be integral into this process. And then we're going to monitor for effectiveness with follow up. So that's a little bit about Legionella. Now let's look at laundry waste and environmental cleaning. So disinfection and sterilization and cleaning are all sort of interchangeably used, sadly, by many, many different people. So cleaning versus disinfection, right? We want to use the terms correctly. So cleaning is literally just the removal of visible bioburden or soil. It doesn't actually kill anything. So I always tell people I clean the toilet and then I disinfect it. Or I clean a surface and then I disinfect it, especially if there's something on it. And you'll see products out there in the United States marketplace that say one step cleaner and disinfectant. What that actually means is that if it's a clean surface, so if I look at the surface and there's nothing on it at all, then yes, I can use that one, wipe or whatever it is and disinfect and clean at the same time. But the reality is, many of our surfaces, especially in a patient care room, do have something on them, right? It may be visible or not. And so we want to make sure that we're always cleaning first, removing that bioburden or soil, and then coming behind that with an appropriate disinfectant. And that's where we actually kill or inactivate the pathogens on the surface. And we have to make sure that we follow the contact time, right? So contact time is the total amount of time that's necessary to kill all the organisms that we're targeting. So let's say that there's 60 organisms on the product's label, and the longest contact time is 3 minutes, right? There may be other product claims on there that say 30 seconds or 2 minutes, but if the longest contact time is 3 minutes, then that's the contact time that we're going to follow, right? Because we want to make sure that we get total microbial kill for the organisms that we're targeting. So again, cleaning, removal of bioburden or soil, disinfection is going to inactivate or kill, right, the pathogens. Now, sterilization is sort of that final approach of let's get rid of everything with the exception of your prions, right? And so when you think about sterilization, it's only as good as it's done correctly and verified. So we can run stuff through a sterilization and it could still fail. And if we don't monitor it, that's a problem. And I give you a pretty sad but dramatic example. And I won't say where it was, but it was here in Atlanta. I got a phone call years ago from a IP risk manager colleague, and they said, we have a situation we'd like a outside counsel for. And I said, okay. And they said, we have a debate here internally. We have an instrument set for the hospital, and it's gone through sterilization. It is past sterilization. So we've passed our sterilization verification test. But when we opened the instrument set, there was a dead gecko lizard inside the instrument set. And I stopped for a second. I thought, did I hear this correctly? And they said, yes, there is. And they actually sent me a picture. I still have the picture. And I said, well, what is the question? And they said, well, there's a debate by SPD as to whether or not we can use the instrument set because from their perspective, the sterilization parameters were met, the sterilization has occurred, and it occurred properly. And the operating room is refusing to use the instrument set because there is considered a foreign body in the instrument set. And the attorney was in the room for the hospital, and I said, I'm not even sure why we're having this conversation. If there is a foreign body, in this case, a dead object, right, in an instrument set, I don't care how sterile it is, right, it is not sterile. But the fact that we had to have that conversation, I thought was pretty alarming. And so sterilization is only as good as it is performed correctly, right? And so we want to make sure that we're going through the steps and the processes. So cleaning, right, always takes place before sterilization as well, particularly with instruments, because we want to make sure that we're maintaining them appropriately. Now let's look on the right side there where you've got Spalding's classification. This was designed years ago, right, to sort of help us identify how will a device or a product be used, and then what is the appropriate level of reprocessing that's needed. Now, it's not foolproof, it's not 100%, but it's about 98% accurate, which is really, really good to hold up for this many years. And it's actually really easy to look at. Critical items are the easiest. So this is going to be a device that's going to be used inside sterile body tissue. So I always tell people if it comes in sterile packaging or it actually goes through sterilization, it is always a critical item. So a catheter, a Foley, a needle and syringe, an implant, instruments, anything like that that goes inside the human body into a sterile body cavity and goes through sterilization, either we do it or it comes sterile, is going to be considered a critical item. Now, a semi-critical item are pretty common, right? And these are ones that come in contact with mucous membranes. So a great example of this would be a flexible endoscope, whether it's used, from the top down or the bottom up. Those are still flexible endoscopes. They require high-level disinfection. Another common example is laryngoscope blades and handles. So those are commonly high-level disinfected. However, we have found that we are not very compliant with that, with laryngoscope blades and handles. And so we've seen many of our hospitals have moved to disposable blades and handles for that reason, because we're not really good and compliant with the safety associated with high-level disinfection. So outside of SPD, it's not something that we really recommend. The other thing, just as an aside, is if you ever see high-level disinfection done in the outpatient setting, you should run, because that is not something that we routinely want to see done. It's just not a safe practice. We're not set up to do that in the outpatient setting. So ideally, that if it's going to be done, should be done in the hospital and the SPD department where you've got properly trained people and the right equipment. The last level there is non-critical items. So these are the bulk of what we use in healthcare, the ones that come in contact with intact skin. So blood pressure cuff, stethoscope, pulse oximeter, thermometers, beds, overbed tables. And here we're going to use a minimum of a low-level disinfectant, but preferably an intermediate level disinfectant. The difference between low and intermediate level disinfectant is really simple. It's Mycobacterium bovis, right? And Mycobacterium, you should recognize as a surrogate to Mycobacterium tuberculosis. Now, we're not worried about TB on a surface, but Microbacterium bovis is a sister organism or surrogate that is very similar to TB, but it is very difficult to kill on environmental surfaces. So that is the difference between a low and an intermediate level disinfectant. And then lastly, you've got your environmental surfaces. And so here we're going to have our environmental services team that's going to come in and really do that cleaning and disinfection. On the bottom left are a couple examples of what we would consider high touch surfaces or priority areas, areas that we know that are touched by us and also touched by the patient, right? Like the nurse call light is extraordinarily contaminated. The overbed table, very contaminated. The TV remote, very contaminated. And we're going to do sort of certain different things during the day, right? If the patient's still there, environmental services is going to come in and do sort of their normal day-to-day cleaning and disinfection. But when the patient leaves, then we have that opportunity to do true terminal cleaning, right, and disinfection. And that's where we have the biggest opportunity to reduce the risk. And keep in mind, if we don't do that well, some patients may get that next bug that was in that room, right, from that patient. And so it's really critical that we resource environmental services appropriately. Now, one question that does come up routinely, and it was also on my exam, I remember, is what are acceptable ways to monitor effectiveness of sort of your environmental services cleaning disinfection program? And we're going to start on the left, which is the sort of poor man's way of doing it, and go all the way up to the right, which is more expensive. So if you think about visual observation, that is me walking around. I watch you do it. As a matter of fact, I was in a building earlier today, and I was really impressed. I was in actually a public building, and they had a contracted environmental services company that was there, and I was blown away at how like impressive this woman was with cleaning a surface. And I actually went out and I told her, I said, who do you work for? And she told me, and I said, well, I am just really impressed with the attentiveness that you have to cleaning and disinfecting all these different surfaces. And she was really appreciative. You could tell no one had ever told her thank you or anything like that. That's visual inspection. I watch with my own eyes, I observe, and I provide feedback, right? Timely feedback. If we want to go to that next level up, then we could look at fluorescent markers, right, where we use UV. And we're going to mark the surface, right? Of course, we don't want to tell the staff which surfaces we're going to mark. And then we come back after they do their cleaning and disinfection process and figure out, right, if that surface has been appropriately cleaned, because then that marker should be gone. That mark should be completely removed when I use my black light. Again, pretty cheap to do that. If we want to go one step above that, we have ATP bioluminescence. Remember, ATP is adenosine triphosphate, which is the living sort of unit of life. And so every cell has it, both living and then recently dead. So it's not going to tell me what is on the surface, but it gives me a sort of digital readout of how much bioburden or soil is present. So we use it as sort of a statistical test. You know, I swab before and then I swab after. So if I swab before and I have 10,000 sort of units, if you will, present on a surface, okay, that's my baseline. And then environmental services comes back and I swab again, and now I have 10. Well, you can do the math, right? Going from 10,000 to 10 is pretty good. But if I have 10,000 and I come back and swab again after cleaning and disinfection and I have 9,500, we need to have a different conversation. So it does give me a more objective measure of, is this successful or is this not? Now, finally, is we've got the plate counts, right? Where we can truly go out and quantify through a microbiological method. This is more expensive, it's time consuming, and it's not foolproof, right? You're going to start to create results. Now, here's what I will mention to you. This is not on the test, but good to know, is if I start culturing things, remember, once I culture, I'm creating data. And when I create data, I have to have a data analysis and sort of approach plan that says, well, what do I do with the data? So if I start to get really bad culture results, risk management and legal are going to say, well, what are you doing about that? So I'm not a huge fan of just random culturing, but I do like a combination of UV, inspection, and ATP. And then lastly is you have what we call direct observation. And this is more where the user, the environmental services professional is watched, they know they're watched, and you have a trained observer that is there doing so. I'll tell you, I don't find a tremendous amount of value in that because when you know you're watched, you typically are going to be your very best. And so I don't think it's a great representation of true practice. So think about sort of the far left ones, those top three ones as probably the best approach. Now, what about actually selecting a product? And I can't stress enough how important it is for the IP to be sitting on the value analysis committee. And if you aren't on the value analysis committee, you should ask to be. You should get training on it. There's even a whole association that has infection prevention specific training called AVAP that you can get involved with. But let's think about how do we select a product from an IP perspective. First and foremost, we want it to be broad spectrum. So gram positives, gram negatives, your envelope, non-envelope viruses, pathogenic fungal organisms, right? We want to make sure it's going to be successful against your bloodborne pathogens and potentially mycobacterium, right? So that's going to get you to that intermediate level and above. What's the contact time? Is it 15 minutes? That's not really helpful. But if it's 3 minutes or 2 minutes or 1 minute, well, that's very doable from a room turnover perspective. So that's very reasonable. Then I've got to look at surface compatibility. Is this something that's going to tear up every surface that I have? So if it's sodium hypochlorite, the number two listed there, that's a bleach solution, right? And bleach is great at inactivation of microorganisms, but it also is not so great on surfaces sometimes. And so it may actually start to devour your surfaces, particularly your plastics. So we need to be careful about that. What about toxicity? What if I use this 50 times a day? What's going to happen to my staff? Do they need to wear a respirator mask? Do they need to wear a particular type of gloves to safely use this? What happens if it gets on their skin? There are luckily no disinfectants on the US market, at least, that are carcinogenic. And so there's none that are going to cause cancer. And the reason they all say to wear gloves, it's not necessarily that the product will hurt you, it's that it's a good industrial hygiene OSHA thing, right? If I'm touching surfaces in the healthcare space, I should not be touching those with bare hands, right? I need to make sure that I'm disinfecting with gloves on to protect myself. Now, that all being said, if I'm disinfecting with a high alcohol product or a bleach product 50 times a day, you're going to get some contact dermatitis, right? Even if you've got great skin. So it is helpful to practice, again, that good industrial hygiene. Then we're going to look for EPA registration status. So in the United States, it is federal law that all disinfectants must be EPA registered. You cannot sell, market, or buy a disinfectant that is not EPA registered. So the way we know that is you look on the product's information, the website, or physically the back of the product, and on the back there'll be two numbers. The EPA registration number, which tells you that the product is registered, aka approved by the EPA. And then the second number is important for us as IPs to know, which is the establishment number. The establishment number tells me physically where that product was manufactured. So if I have a recall, which we're going to talk about in a second, then I have a better idea, right, of where that product was produced so I can understand, well, who do I need to contact? What other product might I need to particularly pull back? And then lastly is I need to think about like cost effectiveness, availability. How easy is it to use? And then what's the environmental impact? The most common disinfectants that are at least used here in the States are going to be quaternary ammoniums, also found in like your laundry detergents. Very, very common. Most quat ammoniums actually have alcohol in them as well to help beef up sort of the efficacy and also the dry time. Sodium hypochlorite is another name for bleach, right? Typically, we're going to see that at a 5,000 or plus parts per million concentration. I always tell people bleach is my go-to for anything, anytime I have vomit or some type of fecal material, because it just is a great organic dissolver and it's highly, highly effective against spore-forming organisms. Then you've got hydrogen peroxide and accelerated hydrogen peroxide. So hydrogen peroxide by itself is very, very bioavailable. And then accelerated hydrogen peroxide is the newer technology that's out there. The challenge that I've at least had personally with accelerated hydrogen peroxide is it doesn't have the best smell. So it's a little bit pungent. It smells kind of like a rotten cherry to a degree. So it's not a great solution for enclosed spaces. And sometimes people will complain about it, but you will see those are becoming more and more common associated with that. Now let's look at laundry and sort of the linen perspective. There are accreditations now, and this is fairly new in the last 10 years for linen services that we strongly recommend. As an IP, you should be visiting your linen facility. whether that is in-house or if you contract. So just because you contract out your linen services doesn't mean that you should not be visiting them. You're still responsible for it, right? And that's true with any of our contractors. So there's healthcare laundry standards that we have that they also have accreditation for where the linen is produced and handled. We want to make sure that we're also meeting our accreditation standards as well. So number one is that when we have soiled linen, right, we want to make sure that's separated. It has to be separated from clean linens. And the big challenge that we have is a lot of nurses will treat all soiled linen as infectious, right? And so we don't want to throw it into the red bag because that's a tremendous cost that's unnecessary. But we do, however, want to make sure we're not handling it routinely. So once it goes into the soiled linen bag, we want to ensure that it's not coming back out, as an example. One of the big things that we see staff do all the time is they'll shake the linen bag. Well, that's aerosolizing all of that nastiness. So we definitely don't want to do that. From a laundry perspective, we want to do a hot water wash with greater than 160 degrees for 25 minutes. Or you can use an EPA registered laundry disinfectant. Most places will do both, right? Because they want to go sort of above and beyond. And then again, CDC recommendations are clean and soiled linen are never stored together. They can be transported together. So if you have a linen service truck, for example, that's coming, they're not going to send two trucks that take, your soiled linen and bring your dirty back and stuff like that, but they have to be separated in the truck. So they should have like a separate compartment where you can have that where they're not going to touch each other or be cross-contaminating. And then from an OSHA perspective, remember this standard falls within the standard personal protective equipment requirements. So let's talk about regulated medical waste, right? That's another piece. This is actually under the auspices of the Department of Transportation at the federal level. So, for example, if we throw away a needle and syringe in regular trash, well, that is a DOT violation. It's also an OSHA violation. So they're going to cover things like your sharps, your pathological waste, your microbiological waste. We want to ensure that if it's sharps, it goes into a fluid resistant, puncture resistant, right, container that is marked appropriately. They're all commercially available. If we're thinking about sharps containers in general, we don't want to overfill those. That's one of the biggest reasons for citations with OSHA. And there may be different ways to dispose. We may have incineration, we may autoclave, we may actually do chemical disinfection. But the big thing with regulated medical waste that we have to be aware of is that while we are not transporting the medical waste, we are 100% responsible for it. So, if you have a bad waste hauler, right, that you've contracted, it does not matter that they are your contractor and they are doing it. You are still responsible. So we want to ensure that we have total confidence in their process and that we're regularly auditing and validating that. That's really important. And so that's typically going to be done by environmental services, potentially your safety officer as well. And that's all part of your OSHA thing. On the left side there, your solid waste, which is typically non-regulated, that is going to also include things like your trace chemotherapy waste, which we have a separate container for that in a different process, your pharmaceutical waste, which that may be regulated by the Drug Enforcement Agency based on your disposable drugs. That might be class 1 and 2. Excuse me. And then you've got your standard precautions for reducing waste in general to make sure that we're disposing of things appropriately in the landfill. Section 4 looks at our recalled or contaminated items. And this is a really big issue. And I've tried to break it down into 4 different categories of equipment, food, medication, and supply. And I want to start with supply first because that is really the biggest one that we see that has the most impact. So those are going to come out from the FDA. So the FDA is going to look at medical devices and say, this item has been recalled. Important to know, though, that recalls do not necessarily originate from the FDA. They almost always originate from the manufacturer of the device. So the device manufacturer will go to the FDA and say, we have a problem. The FDA will say, what do you want to do about it? And then they will typically negotiate a recall of some type. If the manufacturer, for whatever reason, does not play nice, then that is a whole different animal, right? And so if the manufacturer says, yeah, we don't want to do that, the FDA actually has a federal court that is literally in their building, and they can go right down the hall, apply for an injunction with the federal judge, and then they can actually seize the product, seize production. So it's a pretty rigorous process. And they typically do that when the manufacturer is refusing to play ball. So this would be like breaches of sterility, some type of imminent threat to public health. If we look at medication recalls, those are also under the FDA's approval. And so there's different classes of recalls for both devices and medications. There's class one, two, and three. So class three is the one that I don't want to say you don't have to worry about it, but it's the one that you have to worry about the least. Class one are the ones that are most likely to cause serious risk to human health, right? Particularly death, permanent disability. Person's not going to survive this. And so when we see class one or class two recalls, we have a real strict obligation to act on those as quickly as possible. And so if it's drug or medication, those most often go directly to the pharmacy department and they will go to value analysis. And if your hospital has a recall management coordinator, recall management as well. And so we saw a huge outbreak of this with the fungal meningitis, with the compounded drugs years ago that were getting shipped across state lines, which is now why the FDA is involved in this. If we look at the third one, which is food recalls, also under the FDA, but really has some crossover with the USDA from an agricultural perspective and the CDC. So this could be a recall of product that maybe comes into your kitchen and you have to go and recall that from patient trays. It could be in one of your storage areas. It could be something that's not yet served. The recall process says we need to pull it as quickly as possible. We also may have to identify, right, patients that this product was served to. Remember that the patient may have already been discharged. And so that's why it's important to know, well, who got which meals? And that's something that a lot of times we don't think about. And your public health agencies are going to help you with that. And then lastly is going to be your equipment recalls. So again, the FDA is going to do that through the MedWatch process. And we really help in that tremendously by reporting adverse events to the FDA. So remember, an adverse event should be reported not just to the FDA, but also to the device manufacturer at the same time. They're the experts in this, right? We're not the experts in the device. So we want to make sure that they are also in the loop on what that is. And that's done through the MedWatch process, so that you can actually do that online. The great news now is, as of a few weeks ago, all of those adverse event reporting documents are now available in real time. So it used to be months and months and months before FDA would upload those, but now that they're using AI, they actually provide those to us almost in real time, which is pretty impressive. So at the very top there, you'll see our recall response process in general terms, right? Again, we're thinking about general terms for purposes of the exam. The first thing we're going to do is figure out what the alert is, right? So maybe this is an alert for a disposable medical device that has failed sterilization. All right, then we're going to look at the risk level, and they have classified this with the FDA as a class one recall, right? So serious risk to human safety, potential death, potential human disability. Third thing we're going to say is, well, where is it? How many do we have? We have 332 and they're at 53 locations. Okay, great. We need to go quarantine and remove those. And how do we make sure that they're not just left in somebody's office, but they're actually retrieved? The fourth step is we need to think about notification of affected patients and staff. And this is why traceability is huge. Do you have a serial number? Do you know who it was used on? Because if we have 3,000 potential patients, that is very different than 30, right? I definitely want to have 30 versus 3,000. And then lastly is I want to then document what I've done and implement whatever corrections. And the corrections could be that I move to a different product and I say we're no longer using this. But it may not be a situation where I can do that because there may not be any alternatives associated with that. Now let's look at the next section, which is environmental pathogen monitoring. So things like aspergillus, right? One of the quick, easy ways to prevent this is great HEPA filtration, right? We want to make sure we have positive pressure in our rooms. We want to have the ICRA process, which is the infection control risk assessment. And we're now on ICRA 2.0. If you've not had formal training on ICRA, it's a great idea to do so. As a matter of fact, ASHRAE is getting ready, I think maybe May 8th, to offer an online live training on this where you can get certified in it. That's a great thing to have as an IP because then you can look at construction, renovation, and demolition in a completely different way. And they've got the tools that help you with that. What about barriers, right? Are we putting up dust barriers to ensure that there's not cross transmission? Are we monitoring the air and how so? Are we using a certified environmental lab? Remember that most hospital labs are not equipped to monitor the air. That's not capability or technology that they have. So we might need to work with an outside environmental lab that really specializes in that. And sometimes our public health agencies can also help with that as well. And we may need to think about antifungal prophylaxis, right? So if I have high risk patients that I'm concerned about an exposure, I might need to go ahead and proactively treat those. And one of our other reasons that we don't do plants is for this very reason, right? We want to make sure we minimize those. So what about some environmental sources, right? Definitely if we've got construction renovation or we've got tiles, tiles are like the bane of our existence. So maintaining intact ceiling. As a matter of fact, I was in the Atlanta airport the other day and I looked up at the ceiling and I thought, my God, all these tiles are just hanging there. There's no telling what's up in there. Potted plants, as we mentioned, especially in like your bone marrow transplant units, or if you've got poorly maintained management systems, those are always going to be some of the risk if the patient gets this, right, especially a patient with, existing comorbidities, the mortality rate can be as high as 90%. And so this is particularly challenging for us, right? These are patients that do not do well. And our goal here is, of course, to prevent the organism in the first place. What about some of the other things like C. difficile? Remember, C. difficile is a spore-forming organism that forms an endospore. It's really resilient in the environment. It survives well under both heat as well as cold. And they found it for, I think the latest data I've seen was 6 or 7 months with no problem at all. So it is a hardy little thing. And so we typically are going to look at some type of sporicidal disinfectant. It is not just bleach. There are other disinfectants that are EPA registered that are very effective against C. difficile. So you just would look at the product's label in order to do that. But again, we want to use a sporicidal agent to take care of that if we've got a patient with unexplained diarrhea or vomiting. Contact precautions, of course, for these patients. And then we're going to switch out our alcohol-based hand rubs and move to our soap and water so that we get that visible friction rub to remove some of those spores from the environment. What about MRSA and VRE? Right, again, very persistent in the environment, not as much as C. diff, right, because they don't form endospores, but we want to ensure that we're taking care of those critically touched areas and surfaces. So how can we make sure that we do that? Well, good disinfection, good cleaning, monitoring of the surfaces. Are they actually properly disinfecting those? Are we following contact precautions? For our patient perspectives, we may look at skin decolonization, whether that's through bathing with something like chlorhexidine gluconate, mupirocin in the nose is an example. There's all different technologies out there that continue to emerge that might help us sort of get us over the hump, particularly for a procedural thing from that perspective. What about norovirus? Another one fairly closely related to C. diff in terms of sort of transmission risk, right? So this is the one that takes a very small infectious dose, but again, we've got to look at special disinfection sort of approaches. So again, bleach is a great one or another sporicidal agent. My go-to for this is always going to be bleach because we want to stop the transmission, particularly in restrooms. And so it's highly, highly infectious, very low infectious dose required. So here again, we're going to look at contact precautions and droplet during acute illness because it can aerosolize, right? And so we want to try to stop that because we don't have a firm treatment per se for it. It's more of a symptomatic treatment. And then when we look at our last one, which is my favorite fungus out there, sadly, right? This is a multi-drug resistant fungus. And our challenge, particularly in our long-term care environment, is that it is not very easy to treat. And so if you had to pick which one did you want, you always want the bacteria over the fungal pathogens, right? Because when we start to get fungal pathogens, now I'm getting into an issue where maybe my treatment modality is limited. My disinfectant modality may be limited, and my outcomes are not going to be as good, right? So again, contact precautions, which is a theme here, but do you have an EPA registered disinfectant that will be effective against this or others? Another pretty common one that you see on disinfectant products is trichophyton. So trichophyton is common athlete's foot. And again, that's a pretty hardy, resilient organism to kill. So if you're effective against these two, that's a pretty good indicator that you've got a pretty solid product from that perspective. And then our last section that we're going to look at today is construction, renovation, and commissioning. And we sort of stick that under the auspices of ICRA. We are now on ICRA 2.0, which again is coming from ASHRAE, which is the healthcare engineering group. And they provide this matrix, which looks at 4 different classes. So class 1 is going to be sort of your minimal precautions. You can maintain the workflow. And from a dust control perspective, you have to do minimal work. Class 2, we're going to start to put up our temporary barriers. We're going to make sure the adjacent areas are vacuumed with HEPA, and you can have immediate cleanup. All the way up through class 4, right, which is where we're going to look at anti-rooms. You're going to have N95s. And what we do is we use the ICRA framework to determine the classification. And again, if you've not been through ICRA training, go through it with ASHRAE. It's a great program to do. You could do it, like I said, totally online. I think the date is May 8th. You can find it on their website, and it gives you so much knowledge about how do I apply infection control, but to this more specific setting of, the risk associated with construction and renovation. So then you can look at it in the time of, well, is this going to be a major project, maybe something small, right? And you're going to look at sort of the scale, right? And that helps us identify, well, what happens if I do this in the ICU, or if I do this in a HEMOC unit, or maybe I'm doing this in a storage room, right? The risk in a storage room is totally different than if I do this in a burn unit. So that helps us identify what that looks like. Well, what do we do? Well, first and foremost, right, our goal, if possible, and this is always challenging, is to get a seat at the table in design. So, facility designers are special people, we'll just leave it at that, that have their own opinions about everything. And so, if we can, right, we want to get a seat at the table there so that we can be actively engaged in saying, well, here's a great spot for hand hygiene. Here's a good spot for the disinfectant mounts. Here's a great place for the anteroom. Here's where we could put the PPE. If we do that, right, we're proactively designing the environment for the intended purpose, which is a principle of design thinking. Now, on the right side, there are some pre-construction IP things that we can do, like reviewing the scope with our infection prevention committee, conducting the ICRA, which is a requirement for us, determining the type of containment, what types of patients might be impacted, what are going to be our containment measures, right? And how do we measure success? And then look at your construction team. With the construction team, consider that they're contractors, right? But they are contractors that come to your facility. As with any contractor, they have to follow your rules. So that may mean they have to use this product or they have to do this particular step. When they are in your house, they are your guest. And so we never need to be afraid to enforce the rules that we have so that we can ensure the safety of both the patients and the caregivers that we have. And there's a couple of different guidelines that we look for. One of the biggest ones, especially for design, is going to be the Facility Guidelines Institute, otherwise known as FGI. And those are the guidelines for design and construction of healthcare facilities. They're excellent resources, again, more for design and construction. Then you've got ASHRAE 170, which is your ventilation, right? And so this is going to be both for design as well as construction renovation and just general maintenance. So how do we make sure we optimize the airflow for any of our environments? And that's where those air exchanges and the quotas essentially came from before. You do have the older CDC guidelines for environmental infection prevention and control. I personally love them. They haven't changed much and there's not really a reason to, but they have a lot of good meat and potatoes in there. And so that's helpful for us. You do have isolation room ratios for the negative pressure. Remember, we don't ever have enough negative pressure rooms in a hospital. So how do we make sure that we have additional surge capacity is one thing to consider. And then you've got your different accreditation guidelines out there. So what about risk mitigation? Well, there's different things that we can do, right? We can put up barrier systems. We can ensure that we've got a permitting system so that people can't just, do whatever work. We can have negative pressure. We can have HEPA filtration. One of the big things that we can do, though, is to manage the people coming in and out, right? Both who they are, how they function, what are they going to do? Do we have the sticky mats as an example? And frankly, are they appropriately trained? Are these individuals that understand that they're in a hospital and that there's a risk to your patients? And we want to make sure that we're appropriately managing that scenario, right? That's really important for us to do so. If we don't, then we could have potential issues with that. And then from a dust and debris management, this is where HEPA vacuums can come in. Portable air filtration is an example. Covering different things so that debris is not possible to get up into the air are always going to be helpful for us. And commissioning, right, is part of the ICRA process. So before work can start, we want to commission it through the ICRA process. So pre-construction, right, we want to make sure we understand the scope, we understand what's going to happen, what the impact's going to be, anticipated timeframes, and that's where we conduct the ICRA and then we issue the permit. We also want to think about baseline sampling. So if I know I have a water problem today, well, I can't blame that on the construction contractor, right? I need to know that up front so that I have a better idea of what that looks like. Then during construction, of course, we're going to think about, well, is the barrier intact? Do we have proper airflow? Is it still negative pressure, right? What are we doing in terms of our permit? Is it still maintained? Do we need to do a walkthrough? Post-construction commissioning is then going to look at the rebalancing of our HVAC system. Have we done any type of water flushing to ensure that the lines are fully flushed before patients come in? What type of deep clean? And you may even want to bring in an outside service to do that. Contractors, for example, will do what they call contractor clean, which is a very basic clean. It's okay, but it's not going to be the same level of clean as what you expect right before a patient goes in there. You also want to replace the HEPA filter at the conclusion of construction because it's going to be full and contaminated with all the stuff, excuse me, that's out there. And then lastly is your ongoing maintenance, right? Again, we're going to make sure that the HVAC system is properly maintained. We're going to look at our water cultures. We want to schedule out our HEPA filtration, particularly for areas that are really important, like our operating rooms, our bone marrow transplant, our burn units, ICUs, places like that, so that they're properly done. And again, if it's not documented, it never happened. So here's a couple of questions as we wrap up. So the first one is during renovation adjacent to like a hemonc oncology unit or transplant unit, what combination of controls is most appropriate, right? So you've got a couple choices there. And the reason that negative pressure, right, HEPA filtration and dust barriers is appropriate is we want to look at multiple modalities. Right, we want to keep negative pressure so that we're not inviting things, right, extra in. The HEPA filtration is going to give us a higher level of filtration. And then the dust type barriers are, of course, going to keep stuff from contaminating other areas, right? So don't ever select an answer that probably only has one or two things when you have multiple ways that you can hit and attack the problem. What about the second one? An IP is investigating a cluster of Legionnaires. What's your first priority action? Right? With any outbreak investigation, we always want to confirm the diagnosis. Is this real? Is this not? It could be a pseudo outbreak. It could be not anything at all. Just be a cluster of like illness. So we always want to start with confirming the diagnosis. And then lastly is which disinfectant is the recommended choice for C. difficile environmental decontamination. Out of that list, right, it's going to be C. difficile, sodium hypochlorite or bleach, because we want a sporicidal agent. Now, again, there are other sporicidal agents besides bleach. The way you would determine that is to look at the EPA's list. That's not something that's on the exam, but just for your own knowledge. So, a couple of just practice questions for you. So, as we wrap up, have the right HVAC system fully functional, well maintained, and you want it done with a qualified contractor. From a water safety perspective, right, we need to have a program that's run by a qualified individual. Remember, if we don't maintain the water system, we're at risk for things like Legionella, and we want regular flushes. When we think about cleaning, disinfection, and sterilization, know the terms, when they're supposed to be used, and use them appropriately. We always want to clean before we disinfect. And then there's different modalities that we talked about, whether it's walking around, using UV markers, ATP, or potentially culturing. Excuse me. From a recall perspective, start with what is it I need to recall? What's the level of severity? How likely is this to be an issue? Where are the products? Can I get them out, quarantine them, and then document what I'm going to do about it? And then lastly, we talked a little bit about some specific pathogens like C. diff, right? That's going to need a sporicide, or the fungal pathogens, right? That are going to need a specific EPA registered product. Or if I have an immunocompromised patient, do I have a particular label claim on the disinfectant for that? And then lastly, we just reviewed the construction process. So ICRA is at again 2.0, right? That comes from the ASHRAE standards. And the goal is to be sitting at the table during design, but of course that's not always possible. And so if we can help intentionally sort of create the environment. But if we're behind the eight ball or if it's existing facilities, then we need to work with the commissioning process so that we can ensure that there's proper barriers, negative pressure, that there's appropriate air filtration, et cetera. So here's a couple of the references that we discussed today. So I'll stop there and see. Amber, do you have any questions?
Hey, you were mentioning the ASHRAE, I don't know if there's like a webinar or something coming
Yep.
up soon. Is there a way you could email us the information about that?
Yeah, I certainly can.
Thank you so much. But other than
Absolutely.
that, no questions.
Yeah, I'm pretty sure it's that date, but I'm going to actually look right now just to tell you. I
Okay.
I feel like
Because
that's the right.
I looked on their website and they have a lot of different events and stuff. So I was like, I want to make sure I'm looking at the right one.
Yeah, their website is it's a little overwhelming.
A little, yes.
Let's see. So it is, oh no, I was totally wrong. It's May 14th. I was totally wrong. I don't know why I said May 8th, but I will include the web link.
Thank you.
Yep, absolutely. Nick, questions from you.
No questions. I did join a little late, so I'll be looking for the materials a little of this part that I missed, but thank you very much. I appreciate the information.
Yep, no problem. And Robin.
My question has to do with, I know we always tend to lean away from like testing the environment and things like that.
Mhm.
Like my go-to, like starting out kind of fairly new to this, is like, oh, we have this going on. Do we need to start testing this area or culturing this? Like, when do you move to the time where you decide you do want to culture something or move in that direction? Because kind of what I've been told is like, if we test this, we could find something that we're not really, kind of concerned about. So when do you know it's time?
So your second comment is not incorrect, right? I mean, if we test anytime we culture, I've never cultured something that I did not get results that were, we'll just call them problematic. I give you a great example. I cultured an endoscope one time and I got what was the equivalent of pigeon poop. And you may say, well, how in the heck did you get pigeon poop? Well, it was contaminated from the outside through a ledge where pigeons were sitting and the wall was not sealed. Well, that was not anticipated. The hospital certainly did not anticipate that, and that created a whole controversy. And someone almost lost their job over that because they had not communicated to risk management or legal that they were going to culture. And so your point is great. What I would say is if you're going to culture, you should be getting some type of data that you cannot get through any other source that will help you move the needle towards yes or no, this is actually implicated. For example, if you have an outbreak associated with a pathogen and you suspect a medical device is involved, and you've got six patients and they all were, the same device was used on all of them, and you've ruled out every other risk factor, but the device is the only thing that you've not ruled out, then that is a time that I would culture the device. And I would have a plan in place to say, we're going to culture the device. We're going to do it as part as a quality improvement study. Like I'd have a plan in place to do that. But random environmental culturing, random culturing of, I worked with a hospital one time that cultured healthcare providers' hands. And I thought, what in the heck are you doing? And their attorney, I thought, was going to lose her mind when they did that because every healthcare worker's hands were contaminated with MRSA, right? What do you do with that? You can't decolonize all your healthcare workers. So look at the possibility of if I culture this, will it give me information that I cannot get through any other source and will it be meaningful enough to help me say, this is an outbreak, this is not, or give me the information I need to close an outbreak? If the answer to that question is yes, then you culture. But I will tell you 90% of the time the answer is no.
Thank
Does
you
that
very
help?
much. Yeah, it helps a lot. Thank you. I
Yeah.
appreciate it.
Yeah. And I don't ever culture anything without risk management involved, ever. They are my very first phone call, and I always have the attorney involved so that there is a plan in place before I do that. And I will tell you, I don't ever culture people. I did that one time, and I learned a lesson the hard way that that was not a good idea.
Oh, very good information. Thank you.
Yeah. Any other questions from anybody? That was a great question. All right. And just FYI, I know there had been some challenges with the recording in the past. This one was recorded, so you're all set on that. I will send out the recording, the handouts, and I also include the link to the one for next week, which I'm teaching as well, so that you have that, so that everybody will make sure that they're registered, and that will be the last one that we have. But does anyone have any other questions or any issues that Chital and I can help you with? All right. Well, I hope you all have a great evening, and we will see you next week for the final class.
Bye, everyone. Thank
All
you,
right, take care.
Hudson.
All right,
Bye.
absolutely. Good night.